Hi everyone, my name is Érica, and I will present my study about the lineage of foliage-roosting fruit bat, Uroderma. Sorry. Well, Uroderma are genus from Phyllostomidae family, Sternodermatine subfamily, and they are higher distributed in the neotropical zones. They are known as tent-making bats, and they usually eat large fruits. Well, about the taxonomic history, the genus was constantly revised during many years due to the difficulty in split the species, and during part of its history, the genus was reporting have only two species, Uroderma magnificarum and Uroderma bilobatum, and the last one was divided in 5 subspecies according to the geographical distribution, and after some years later, the 60 subspecies was described according the diploid number. Well, recently we have sorry about that because so recently at the 2014 we have a new study using molecular data which divided the species in 5 species, a new species that described recently called Uroderma bakeri, Uroderma magnificarum, and Uroderma bilobatum with 2 subspecies, Uroderma convexum, which was a subspecies before and now is a species, but also have 1 subspecies, and Uroderma davisi, which was a subspecies, now is considered an species. Well, although the new classification, we still have some issues to discuss, like the chromosome races and the contact zones. The chromosome races is something that was described at the 70 decades, which was 3 chromosome races for the complex Uroderma bilobatum, which was represented according to the geographic distribution. And this is the other species that from Uroderma, which have the diploid number equals 36. And between these 3 chromosome races, we have a contact zone and the Central American. These contact zones between the 2 species, which have the diploid number equals 38 and 44. This species, these individuals can have hybrid individuals, and we also identified some integration process. So to these informations, we decided to check the Uroderma status using molecular and morphological data. For this, we did this sample. We sample all the distribution of genus Uroderma, and we choose at the red squares are data from the molecular, and the blue squares are the morphological data. So to DNA data, we used 2 mitochondrial genes, the Cytochrome B and CO1. Also, we did the phylogeny analysis as maximum likelihood, Bayesian inference. We did divergence time. We did the reconstruction of the chromosomal races, and some population analysis as BAPS and Arlequin. For the morphological data, we did 9 skull measures, 29 discrete characters as color fur or the shape of the teeth. And to analyze all those data, we did the principal components analysis. And this is the results. We have a phylogenetic analysis, which rescue 5 clades, a clade of Uroderma bilobatum, a clade of Uroderma convexum, another of Uroderma davisi and Uroderma bakeri, and Uroderma magninihostrum. So the time of divergence indicated that this genus diversified during the late Miocene, followed by explosive diversification, because we have similar mean dates of the 5 clades. We have the oldest clade, which was Uroderma convexum, but this species seems like they diversified at the same time, almost the same time. So during this period, the South America landscape changed completely. We have some events like uplift of the ends, the basin formations, and the climate change. All these events might have triggered the rapid diversification of some of groups of the Uroderma in a short period, while it conserved some phenotypes, which the wide range of the distribution in Uroderma across different biomes and ecosystem can hide higher diversification and population organization. So, and when we verify the clades inside of each large clade, we only verify the 2 clades very well supported at the Uroderma bilobatum, which we choose to do the population analysis. The population analysis show us to structure population, and in the eastern Amazon and the western Amazon, we have 2 different populations, and some of these individuals can how can I say, they can mix it with another population. As this example of individuals, there are mixing individuals with both populations. And also, we did a reconstructure chromosomal race, which was, we used the molecular data and the information that we have about the diploid number, and what we find, it is the possible ancestral number is 36, and we have followed by a split from the 42 diploid number, and what we have as a new information, as we supposed that the new species Uroderma bakeri have the 44 diploid number. This species is a new species that was described recently, and we don't have any information about the diploid number. So we suggested that it's similar to Uroderma davisi. Do the molecular data show us that they have some similarities? Maybe due the fact that these 2 species live in a higher elevation, yes, it's higher elevation. So about the morphology, we didn't find any sexual dimorphism, which was expected. And what we saw that it was interesting, that we have 2 species, Uroderma davisi and Uroderma convexum, that are very different species in the molecular data, they position in the clay in the phylogenic tree is very different, but when we saw the female morphology, we saw some similarities. And what we think about this is that these two species are the species that can make some hybrid individuals. They live in the Central America, and they can do hybrid individuals, and we have integration between these two species. Maybe this is because the similarity of the females that we find. So, and also we didn't find any discrete characteristic that was very strong that we can use to define the species. So this discrete information was very have a lot of polymorphisms. So and finally, what we saw in this study is that for the first time we saw some population structure about the bilobatum, the Uroderma bilobatum. We also saw that the discrete data is not a good data to divide split these individuals in species. We also didn't support the subspecies status due to the fact that we didn't find any informations in the molecular information. We can have any support about this status, and we didn't find any support of this status for the morphological data. And finally, what we have in this study is that the fact that the Uroderma genus is a very interesting group that we can have an example of very fast diversification that we can see a lot of information. Like this diversification give us a diversification of diploid number and some, but not diversification of some morphological data. And one thing is very important, that for the first time we also have another individuals from Uroderma bakeri, that this species was described recently, and we have only one sequence, but in our study we find another sequence from this species. So I would like to thank you, the audience. I would like to thank all these institutes that give us some samples of DNA samples, some tissues, that give us the opportunity to visit it, and gave me the opportunity to analyze the morphological data. Also, I would like to thank the INPA, that is Institute from Amazon, and CAPES and FAPEA that gave me the grants during my master degree. So thank you very much.